Buongiorno. Here's our review. Let's begin. All right, so the first one says, describe each of the following genetic disorders. What you need to make sure that you know here when you're talking about it is Tay-Sachs, sickle cell, achondroplasia, Down syndrome, Huntington's disease. 1 and 2 really kind of go together. You want to be able to talk about the genetic disorder and what is Tay-Sachs? What would it look like in its karyotype? What causes it? And so you want to know the details. We're not talking about like geneticists, you need to know that kind of stuff, but Down syndrome, trisomy 21, you want to know things like that. And if you looked at the karyotype, so if you're looking at a karyotype, let's just use Down syndrome as an example, you're going to see 3 chromosomes at chromosome 21. So, you know, something simple along those lines, something very simple as we go through and look at the karyotype. All right, number 3 says, and then things like describing it. For example, sickle cell. Sickle cell anemia is a homozygous recessive, has a homozygous recessive allelic combination on the autosomes. Whoa, what does that mean? Homozygous recessive, so you've got 2 recessive alleles, 2 lowercase, and they combine and those are on the autosome. They are not in the sex chromosome. So 1 through 22 is where you're going to find sickle cell. All right, what is a tetrad? So if we look over here, whoops, that's not the one. If we look up here to the pictures, that's a tetrad. Tetra means what? It means 4. So this is a tetrad. And what it is a 4-part structure that's going to form during prophase of meiosis. And it consists of 2 homologous chromosomes. So here's 1 homologous chromosome. Here's the other homologous chromosome. And each homologous, each homologous chromosome is composed of 2 sister chromatids. That's just a regular definition of a chromosome, of 2 sister chromatids. But now with a tetrad, you have 2 sets of those. All right? Ding, let's go back here. All right, what's the difference between inversions, translocations, and deletions? From our activity yesterday with disorder detectives, clearly we were able to see that an inversion is part of the chromosome, breaks away, and reattaches to the same chromosome. That's the key, it's on the same chromosome. Where when you have a translocation, you have part of the chromosome that goes from part of one chromosome then goes over to another chromosome, all right, in a translocation. And inversion's on the same one, so you have the movement of the genes on that chromosome. Translocation, you have a segment of those genes that travels to another. So with a translocation, you'd see one that's larger and one that's smaller in there. And then with a deletion, it is just as it says, part of the chromosome or a part of that sequence of DNA is deleted. So let me show you here a picture, possibly, if I can. Oops, I got to get out of this mode. Here we go. All right, there we go. All right, so if we go down here, you can see this is a deletion. So this is before the deletion, and then this is after the deletion, because the deletion, you can see how this is shorter. And then here, with an inversion, here, this is just a paracentric one. It's just an example, but this region is now up here. So you'd see the banding would look different, and they're just inverted. But it's still on that same chromosome. This isn't going to here, and this isn't going to here. This is the chromosome, and then that on the other one, it's inverted. All right. And then over here, we have our translocation. So we have this information going over to here. So we see this area here going on over, and you can see the switch there. All right. Describe crossing over and its importance. So crossing over, if we look at this picture right here, There we go. Okay, so here's crossing over. And it's going to occur in prophase 1. And we have 2 chromosomes cross over and exchange their genetic information. So here's the tetrad. And you can see where they cross over here. So we have blue crossing over red. Those aren't the real colors of the chromosomes, but we do that so that we can demonstrate what's happening. All right, and so, and then you have, in metaphase 1, you start to have these pulling apart at the kinetochore, at the centromere there. And then you have, and you can see when these are pulled apart, now you have red tips on the blue chromosome and vice versa. So this genetic material from this one is now over here. So not only are you getting chromosomes from mom and dad, are you getting information from mom and dad, but now it's even within mom and dad, now we're mixing everything up. And ultimately, we end up with the 4 haploid cells, and these 2 have more genetic variety to them. All right? Okay. Oh, I gotta look at my timer here. I don't know how much time. Okay. Now we're moving on here to number 6. What gametes are produced from a... the other thing, sorry, going back to number 5, its importance, the genetic variety. We need the variation. Remember, we've talked about this several times, where we've talked about why that variety is so important, especially, I mean, when we talked about asexual versus sexual reproduction. We need the variety, because if one disease comes through and it's going to wipe out one, it would wipe out everyone. So as far as survivability, the more variety there is, the higher the survivability rate. All right, let's go on to 6 now. What gametes are produced from a heterozygous genotype, homozygous dominant genotype, and a homozygous recessive genotype? Well, ultimately, if we have a heterozygous, that's a capital letter and a small, dominant and a recessive allele, and you want to look at what is produced. So if we have a heterozygous crossing, we're going to get, well, from heterozygous, one allele is going to be a capital letter, a dominant allele. The other one is a lowercase. That's going to be a lowercase allele. So with heterozygous, we're going to end up with big P, little p. So one allele's capital, one allele's lowercase. With homozygous dominant, both capital letters. And with homozygous recessive, obviously one allele, that's a little p, we'll make that a little flippy thing there. One little p and another little p. All right? Okay, away we go. Number 7, red-green color blindness. If we're describing this, we are looking at, you want to look at and describe everything related to red-green color blindness. Why do we have it? And be able to understand the Punnett square responsible for it. So when you're looking at red-green color blindness, first of all, you're probably going to notice that it's going to be a sex-linked trait. And you're going to follow it. Just make sure that you can work the Punnett square related to red-green color blindness. All right, number 8, the phases of meiosis. All right. I had it in here. Where did I put it? Huh. I don't know where I put it. Okay. Let's skip that. I'll come back and I'll find it and I'll put it in the next video. All right. Sorry about that. La 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 la. Where's the next question? Where are alleles found? Where are they found? Well, they're found on homologous chromosomes is where alleles are found. Number 11, what is the purpose of meiosis? Well, we have to have a reduction in the number of chromosomes. We can't have, in the chromosome number, we can't have 46, because if we had 46 sperm, 46 chromosomes in the sperm and 46 in the egg, we would end up with 92, and that would not be a viable human. And so it would not survive. And so we have to have this reduction in the number of chromosomes, and we also have the variability. We end up having a set of cells that have a very unique set of genetic material. What is a homologous chromosome? Homologous chromosome is a set of one maternal chromosome and one paternal chromosome. They pair up with each other inside a cell during meiosis. So you have one from mom, one from dad. They pair up during meiosis. Genes like big M, little m are located on homologous chromosomes. So big M is one allele and little m is another allele of that gene. Okay, lastly for this video, difference between law of independent assortment and the law of segregation. Law of segregation is Mendel's first law. All right, so it's his first law, and it states that there is a pair of alleles for each trait. And independent assortment is Mendel's second law, and it is also known as the law of inheritance. And basically what he says here, he further states that alleles sort independently to form a gamete. In other words, the allele of a particular trait does not have any effect on any other alleles during the formation of gametes. So they do not, I mean, just think of the name. Law of segregation, they're going to separate. And law of independent assortment, they are going, they're not attached to each other. They're not connected to each other in a sense. They are, they will sort independently. So segregation describes that you're going to have these two alleles for a particular trait, and those are separated during gametogenesis. So when haploid gametes are formed. On the other hand, the law of independent assortment describes that those separated alleles for different traits can combine into haploid chromosomes in any combination. All right, let's stop there and we'll move to video 2.